Good evening and welcome you all to this new course which will be starting today on cell culture technology. So, the name cell culture, whenever it comes across, people have the first feeling that, well, it is a lab training or a kind of a technique which is used by most of the labs who work at the interface area of biology, chemistry, environmental sciences and all other allied fields, biomedical sciences, say for example. So, it is just a simple technique what most of the laymen kind of understand, that cells which are cultured in an synthetic or artificial environment. At least that is what the commoner, or you ask any graduate student or undergraduate student, that is what they will answer. Why we really want to offer a course in this area? So, and as a matter of fact, when I floated this course, there are people who ask me that is a practical training. How really you want to put across a course which is mostly a practical training? People have cell culture labs. So, for me to put it across is, though the name is just cell culture, but there is a philosophy behind this whole thing. There are some basic rules, and moreover there is a tremendous amount of science and art involved in it. And if somebody who wants to do cell culture or wants to learn cell culture appreciates these basic philosophy or the paradigm or the milestones, then it will be easy for that individual to design the problems better, and most importantly to appreciate how far they can go. Say for example, in mathematics there are several techniques, right? Or in physics there are several techniques. Similarly, in chemistry there are several techniques, say spectroscopy technique. But we have course, the basic logics behind a spectroscopy, whether it is a Raman spectroscopy, whether it is a FTIR, whether it is NMR. If you know those, at least the basics, then you know with your problem what all you can derive using spectroscopy, or say for example crystallography. Similarly, several mathematical tools, similarly like statistics, these are tools. But if you know the tools and the origin of the tools and the power of the tools, it will help you to become much more wiser in designing your problem. Similarly, cell culture is a tool, but tool whose journey, if we look back, is now more than 100 years old. And there are a handful of books you will come across, and some of them I have already mentioned, like Ian Freshney's tissue culture book, or few other books like Gosselin Banker's neural cell culture book. And there are manuals published by companies like Jipco, Invitrogen, or currently which is known as Life Science Technologies. And they are really nice guide. But one thing which I personally have felt is that somewhere these books are more or less like a protocol book, but cell culture as such is not just a mere protocol. Yes, definitely there is a significant part of it which is protocol driven. Just like in molecular biology, you have Maniatis's manual, you can go through it, okay. But still molecular biology as a subject has tremendous amount of chemical logics And unless you understand that, you will be working like a blind person. That is precisely what happens in cell culture, that a young student or a young enthusiastic undergraduate or a postgraduate just embraces that this is technical, just let follow it, without putting logics in place, that why am I doing it? What is the basic reason behind it? So, in this course, what we will try to do, of course, we will talk about the techniques, but mostly we will be talking about the philosophy and what drives us and what are the possible options, some explored, some unexplored. As I mentioned just a couple of minutes back, the history of cell culture is now more than 100 years old. And if you look back, like if you are a good historian or if you are interested in scientific history, if you look back, the very first paper which at least known to us, which were published in this field were around 1908, 1912. It is that back, even much earlier than the First World War. First World War started in 1914, right. So, this was the paper from Harrison, one of the pioneers, and what he did, for the first time he grew explant. Explant means part of some animal system's body on a matrix, and the matrix was a very interesting thing. It was a spider net. He did not know where to grow. So, if we break up this problem, what he was trying to do, but before what he was trying to do, let us go back a little. So, if this field, which has its first set of publication coming almost 117 years back, then foremost what we will try to do, we will try to appreciate this whole history of 100 years, what has happened, how things have progressed, and where this can take us. So, there are points, and it is not that in just 1 or 2 classes we are going to finish this off. As we will be proceeding through the course, time to time we will try to go back and see, where it all started. That will kind of give you an idea that where to go. If you do not know the history, if you do not know the time, how it has been progressing, it is very tough to build up a story, what will be the next landmark thing which is going to come up there. So, on one hand, when I started the lecture today, I told you that this is used as a technique for certain people, yet there are many people who have spent their lifetime in or devoted their lifetime in discovering or making this field to march ahead. So, keeping this in mind, let us go a little bit back to 18th century, when the cellular theory which was given. So, one of the theory was omne cellulae cellulae, means self existing from pre-existing cells. Here I will just take a slight detour for you. Course title is Cell Culture Technology, and in this particular next series of 40 lectures, we will be exclusively concentrating on animal cells. Okay. This is just for your note. We are not taking plant cells into consideration here, because that will become way too diverse. We will talk only about animal cells. So, cell culture technology, you can put it within bracket animal cells. Okay. And of course, we will be talking about all sorts of animals, starting from mammalian cells, even human cell culture, to mammalian cells like rats, mice, all the way to amphibians like lower order, including fishes. Okay. This is the range, the spectrum what we will be covering. Okay. Coming back to the basic fundamentals. Cell arises from pre-existing cells. So, that means each of our cells in our body arises from the previous one. If this is true, then could we do this thing outside the system? What does that mean? That means, say for example, I have these epidermal cells growing all over the skin. I take a small part of it. So, what I will be taking out will be a small tissue. So, all of you are aware about the organization. You have cells, cluster of cells performing a common function is called a tissue. I take a tissue. So, each cell is adhering to the other cell with a cementing matrix. I dissolve the cementing matrix. So, what I have are individual cells, and if I take this cell outside the system, so no more in part of my body now, and plate it somewhere, will it behave the same way as it happens in my body? Is it making sense? Let me just put it now graphically for you to appreciate it. Okay? So, to start off with is our cell culture technology. Okay. And we will be dealing, as I mentioned, exclusively with animal cells. And what I am trying to tell you is that, say for example, from an animal, I isolate or dissect a tissue. Now, once I dissect this tissue, so it will be something like this. For example, there is a mass of cells which I have isolated. Okay. Now, once I isolate this, I have two options. Either I just grow them as it is, which we will call as outside the system. Now, up to this was all inside, which is in vivo. So, there is a term which is used for this. If you are directly studying within the animal, it is called in vivo. Now, I have in vitro conditions, which is outside the system. Now, I directly grow this piece of tissue, growing the intact piece of tissue outside the body. In that situation, I will call it a tissue culture. Of course, if this is a dividing cell, then I will be expecting, my expectation with this in a tissue culture dish will be, these are the old cells which are present, then going by the theory of omni cellulae cellulae, cells existing arriving from pre-existing cells, I should be able to see the development of the new cells, the one which are shown in red. So, they will be dividing and they will be forming a mass kind of a structure, right. These red ones are the ones which are growing outside the system. Or what I do, I dissect this tissue and I follow another route where I dissociate the tissue into single cell suspension. What does that mean? Let me suspension. Okay. That means, so I had this tissue here, okay, the original tissue which I took out from the animal. So, this tissue has multiple cells and each one of these cells are anchored to each other, where I am putting this small red stumps. These are the extracellular matrix or the cementing material which are making these cells to adhere to each other. Okay. Now, what I do, I take some kind of an enzyme or some kind of a system by which I break down or chew away those cementing materials. So, let me introduce that. So, I introduce some form of a compound which will start to nullify them. Okay. So, once this cementing material is dissolved, what I will be leaving behind will be, after this, let me put, this is the cell cementing material, and I am not introducing any technical terminology at this point. We will come later on to all those things. Cell cementing material, and so these are compounds dissolving cell cementing material. What we will be getting after that will be a suspension single cells like this, which are no more adhered to each other, they are suspension, and this is called the word which I used in the previous slide, single cell suspension. So, this is called single cell suspension. Now, if this single cell suspension is given a right environment to grow, say for example, in a dish or somewhere, and okay, let me introduce this complexity, let me finish this and then I will introduce this complexity. What is that and where I stopped? If I allow them to grow on a synthetic environment, then they should be able to exactly behave like, they should be able to divide like this. So, this green is showing they are dividing, which is proving the point omne cellulae cellulae. What do you say it? Cells arising from pre-existing cells. Okay. Now, second situation what we told you is called cell culture. The first one is tissue culture, second one is called cell culture. So, there will be these two words have been used very haphazardly in the literature, because when Harrison, I told you that 1903 the first paper by Harrison, when Harrison did, actually what Harrison did was something like a tissue culture. He took an explant, that is why it is called an explant. He took an explant or part of the tissue and grew it on a spider net, and so it was, I think it was a neural tissue, and it shows the extension of the neural cell processes moving along the threads of the spider net. Okay. So, essentially the term which is coined was tissue culture, but over a period of time what we will be talking mostly about cell culture, because the reason is there are different terms for all these things. When you start with single cell, you allow it to grow to form a mass or form a tissue, there is the next level. If you allow the tissue to go in a 3 dimension, that is called explant. Sometimes people directly get the explants. So, here we will be dealing at different level. So, to start off with, in order to, since the course title is cell culture, so I told you this is purely what is the cell culture, but we will be covering the whole spectrum from cell culture to tissue culture to explant culture, all those things will be covering. But for the basics, it should be very clear to you what you are doing. And now we are into 21st century. So, we should not use the wrong term, because it is not the wrong, it is just the confused. So, it is better to use the right terminology. What am I doing? I am doing cell culture. Am I doing tissue culture? Am I doing explant culture? It should be very clear to you. And there should not be any room for any ambiguity. Now, coming back. If it is omne cellulae cellulae, cell arises from the pre-existing cell by Rudolf Virchow when he proposed this, which is part of the basic principles of cell theory. Now, the question is, yes, indeed, what here when I am drawing that yes, from the pre-existing cell I am getting another cell. Sure. Sorry, I mean out here. Yeah, definitely from cell, this green one, this is a new cell. Now, the question is how much closely this new cell which arose from the pre-existing cell is similar to its parent. So, if this one is the parent, this is the parent cell, okay? This is the parent cell, and this is the progeny cell. Yes, they look similar, but are they truly similar in terms of structure? So, if I had to compare between these two, what I will be comparing is structure and function. Are they same? Not only that, are their structure function same as their in vivo counterpart? What is the in vivo counterpart? Here are these cells. Say for example, if I just add a little bit more here, like if I talk about the cell culture here, if these individual cells which are growing instead of the tissue, and if these are their next generation forming, are these cells, properties of these cells, are they equal to the properties of these cells? If it is so, there are two options hypothetically. One, they all behave exactly the same way. Behave the same way means all their expression profile of different proteins, all other parameters are same, or being outside the system, because when they are inside the system, any cell which is growing here, it is exposed to blood vessels, it is exposed to other tissue, it is exposed to n number of things. But the very moment I take this part or any part of the body and growing it outside the system, it is in a totally different environment. It is a totally synthetic, totally different thing. It may do many bizarre things which inside the body it cannot do. Say for example, a cell which is growing here, it is under normal control of several parameters, it cannot, there won't be any outgrowth or anything. But same cell, there is a possibility, if I take it out and grow it outside, it may behave like a cancerous cell, it may behave like something else, which we have no clue, because it does not have any restriction boundaries, it can grow. So, how close, when we are growing something outside the system, how close we are to the in vivo setup, because that will determine a lot of things, that how, what you are interpreting, your experimental interpretation of using a technique based on this, and you wanted to extrapolate and claim that this is what is happening in an animal, will depend a whole lot on how close these two systems are, and if they are not close enough, how far they are. So, say for example, now let us draw the lines. Okay. So, what we will be doing in the next class, we will start to draw the lines that how close are these cells to their in vivo counterparts. Okay. So, let us close in here for the first class, and we will go to the next class where we will be discussing this similarity and dissimilarity, and we will come to a conclusion that where all the real challenges lies. Okay. Thank you for a patient listening.